Genetics is the study of genes, including the structure of genetic material, what information is stored in the genes, how the genes are expressed, and how the genetic information is transferred. Genetic is also the study of hereditary and variation, the arrangement of the gene within organisms is its genotype and the physical characteristic and organism based on its genotypes and the interaction with its environment make up its phenotype. Exchange of genetic information in bacteria, mutation in bacteria. Mutation arise in bacterial population, induced spontaneous. Rare mutation are expressed. Bacteria are haploid. Rapid growth rate has these characters. Selective advantage in reach from mutant. Gene transfer occur in bacteria. General features of gene transfer in bacteria. Unidirectional, donor to recipient. Donor doesn't give an entire chromosome. Merozygotes. Gene transfer can occur between species. The transformation, gene transfer resulting from the uptake of DNA from a donor. Factor affecting transformation, DNA size and state, sensitivity to nuclease, competence of the recipient, Bacillus Haemophilus, Neisseria, Streptococcus, competence factor, induced competence. Transformation steps, uptake of DNA, gram positive or gram negative, recombination, legitimate, homologous or general, significance, phase variation in Neisseria, recombinant in DNA technology, transduction, definition, gene transfer from a donor to recipient by way of bacteriophage or phage, a virus that infects bacteria. Phage composition, nucleic acid, genome size, modified based protein. This is the head, this is the tail, contractile sheath, this is the tail fiber, the base plate, structure of T4, size 80 100 nanometer, head or capsid. Infection of the host cell by phage, adsorption, tail fiber, receptor in LBS, invisible attachment, sheath contraction, nucleic acid injection, and DNA uptake. Types of bacteriophage: lytic, virulent, phage that multiply within the host cell, lyse the cell and release progeny. Phage, lysogenic or temperate phage, phage that can either multiply via the lytic cycle or enter the quiescent state in the bacterial cell. Expression of most phage gene repress prophage phage DNA in the quiescent state, lysogen bacteria harboring a prophage, event leading to circulation of the phage chromosome, cohesive ends, linear double-stranded, open circle, ligase, closed circle. Event leading to lysogeny. This is the stage. Site-specific recombinant phage-coded enzyme. Repression of the phage genome. Repressor protein. Specific immunity to the superinfection. Termination of lysogeny. Induction. Adverse conditions. The role of protease. Destruction of repressor. Gene expression, excision, and lytic cross. Transduction. Definition: gene transfer from a donor to recipient by way of bacteriophage resistant to environmental nuclease. Transduction. Types of transduction: generalized transduction, in which potentially any donor bacterial gene can be transferred generalized transduction, infection of the donor, phage replication and degradation of the host DNA, assembly of the phage particle, release of the phage, infection of recipient, homologous recombination. Potential any donor gene can be transferred by bacteriophage. Transduction, types of transduction. Generalized transduction is in which potentially any donor bacterial gene can be transferred. Specialized transduction in which only certain donor gene can be transferred. The definition of transduction, types of transduction, significance, common in gram-positive bacteria, lysogenic phage conversion, Crinibacterium diphtheria conjugation. Definition, gene transfer from a donor to recipient by direct physical contact between cells. Mating types in bacteria. This F factor, fertility factor, F6 pilus, the donor, and this is the recipient. Lack of F factor, recipient, lack of F factor. Physical states of F factor, integrated HFR, characteristic of HFR X, F crosses. F rarely become HFR while HFR remain HFR. High transfer of certain donor chromosomal gene. This is the F factor. Physiological state of F factor, autonomous with donor gene. Characteristic of F crosses, HFR, F become F while F remain F. High transfer of donor gene of F and low transfer of other donor chromosomal gene. Mechanism of F plus, F minus. Pair information, conjugation bridge, F plus, F minus. Here the transfer F plus, F plus, F plus, F plus. DNA transfer, origin of transfer, ruling circle replication, conjugation, significance, gram-negative bacteria, antibiotic resistance, rapid spread, gram-positive bacteria, production of adhesive material by donor cell, transposable genetic element. Definition, the segments of DNA that are able to move from one location to another. Properties, random movement, not capable of self-replication, not a replicant. Transposition mediated by site-specific recombination. Transcription may be accompanied by duplication. Types of transposable genetic element. Insertion sequence, elements that carry no other gene except those involved in the transposition. Nomenclature and structure. Importance, the mutation, plasmid insertion, phase variation. H1 gene, H2 gene, H2 flagella, H1 flagella A, H2 flagella A. Plasmid definition, extra chromosomal genetic elements that are capable of autonomous replication. Episome, plasmids that can integrate into the chromosomes. Classification of plasmid, transfer properties, conjugative, non-conjugative, phenotypic effect, fertility, bacteriocene genetic plasmid, resistant. Structure of R factor, RTF, R determinant, conjugative plasmid transfer gene, R determinant resistant gene,